Hi everyone, welcome to my channel Pure Biology, and today I come with another video that is related to immunoglobulin, okay, or antigen antibody. So immunoglobulin, today we will learn the structure of the immunoglobulin, okay, the structure of the immunoglobulin. And here this is the rough structure, very rough structure for just showing these features with this antibody molecule, a antigen molecule or immunoglobulin, not antigen molecule, an immunoglobulin molecule. And this is the rough structure for showing this expansion. Okay. So let's continue. This is first one. This is Y-shaped molecule. You can see that this is Y-shaped molecule, okay, and two identical binding sites. Two identical binding sites. This one and this one. Clear? One at the each tip of the arm. So if you consider these are the arms, so this one is the tip of the arm. That is the N-terminal arm. You can say this is the N-terminal arm, and this arm is actually the antigen binding site or ABS. This is the ABS site. Clear? This molecule is bivalent. Why this is called the bivalent? Because there are the two arms. So this is the bivalent. And number two, this has a common structure of 4 polypeptide chains. Here are 4 polypeptide chains, means 2 light chains and 2 heavy chains. Okay, 2 light chains and 2 heavy chains. So 2 light chains, generally that light chain, one light chain that consisting of the 200 amino acids, and heavy chain that consisting of the 440 amino acids. Okay, so why this is consisting of the 2 chains, that are the 2 L chains and 2 H chains, L for light and H for heavy chains. It is polypeptide. It is called the consisting of the polypeptide chains. Next, each L chain is bound to H chain by the disulfide bridge. Okay, disulfide bridge means SS bonds are there. These 2 chains are connected with each other or bound to each other. Here also SS bond, here also SS bond. These 2 chains are bound to each other by the SS bond. Okay. Next one, the each chain and obviously some non-covalent linkages and some non-covalent linkages. 5 different chains generally we can see in the immunoglobulin molecule: alpha, gamma, delta, epsilon and mu. Clear? And these chains, after these showing or these understanding these chains, we will just entering or we will go inside of this molecule. Here we will get the domains. Domains mean L chain that consisting of variable domain VL and one constant domain. So what domains? Each L chain, it has one variable domain, that is VL, L for L chain, and one constant domain CL. You can see here VL, that means this one is the VL is the variable domain, and CL, that is the constant domain. So this is the L chain. You can see this is the L chain. Here you also see that CL, that means constant domain, and VL is the variable domain. And in case of H chain, this has one variable domain and 3 to 4, one variable domain and 3 to 4 constant domain: CH1, CH2, CH3, CH4. This is VH. Starting the constant domain from here, CH1, next CH2, CH3, and obviously in the same case here, the variable domain is the in the heavy chain, that is the VH, VL, not VH, VH, and variable domain VH, and after that CH1 and CH2 and CH3. So CH1, CH2, CH3, these are the constant domain, and VH is the variable domain of the heavy chain. So this is the heavy chain. Okay, these two are the heavy chains, and these two are the light chains. And heavy chain and light chains both are connected or bound with each other by the disulfide bonds. Two disulfide bonds, di means two S bonds, and obviously some other linkages, covalent linkages. Next, in each VH and VL domains, variability of amino acid remain restricted to a number of small hyper variable regions. This is called the complementarity determining. This is the determining, okay, determining region, okay. Complement binding site. This is called the complement binding site. This is the FC receptor binding site or the complement binding site. Or you can say that this region is known as the complementarity determining region. Why this is complementarity determining region? Because here VH and VL domain, in that the variability of the amino acid is seen. That means if you consider one, just distinguish one immunoglobulin from another by seeing the variability of the amino acid, and this amino acid variability is seen where? This region, complement binding site, this region. Clear? Okay. Next we I have to rub it and then explain again. Okay, so here two other region we have to know what are these. That is the FCR region or framework region. Okay, framework region. Here the variation is less seen. Okay, less variation is seen. And FAB and the first region is the complementarity determining region. Okay, that means here the number of what is that? The variability of the amino acids is seen here. And another region, this is called the FCR region, where the framework region, there the variation of the amino acid is very less. Okay. Now, what are the FAB and FC region or fragment? You can say the two identical fragments. These are the FAB and FC. You can see here. This is FC and this is FAB. Okay. This region. So, this FAB region actually is the antigen binding site. Okay, and here the antigen can binds, antigen binding sites, and the rest is called the FC, that is the not binding site. Which one? This is the FC region, this is the not binding site. The first portion, that is the antigen binding site, and the second portion, this is the FC region called the not antigen binding site. Clear? That means there is the actually facility to bind with the antigen with the antibody, but this is the place where there is no chance to bind the antigen with the antibody. Clear? So that's why the first portion is known as the FAB region and the second portion is known as the FC region. Clear? And another thing, this is happened only in the splitting of the immunoglobulin or the antibody when it's possible, if there is any enzyme. Here I name one enzyme, that is the papain. Okay, this enzyme, suppose. So any enzyme is there, or you can imagine that papain is there. So that means the antibody molecule is totally splitted, and here the two regions are formed. Clear? One is the FAB region and another is the FC region. And now the two functions we have to know. What are the actual functions of the variable domain and actual function of the constant domain? The variable domain actual function, that is, it determines the wide range of antigen binding specificity. Okay, specific antigen may bind with the specific immunoglobulin, and this is possible due to presence of the variable domain. Clear? And what is the function of the constant domain? This actually extends the FAB arms. This FAB arms is extended by this constant domain, and why this extension is needed? That means more antigen can bind with the antibody molecule or the immunoglobulin molecule. Okay, so maximum interactions of the FAB arms with the antigen molecules. Okay, so this is the very brief structure of the immunoglobulin, and this is my first topic related to immunoglobulin. So please do continue and share my channel with your friends for getting the knowledges that obviously from the biology, not specific part that is including genetics, immunology, environment biology. So continue with my channel, and obviously you check out my previous all videos. I have already explained different topics related to the biological science, so please you check out that one. And if this is very helpful to you, so please subscribe and share with your friends. Okay, thank you.